So the presentation delved into the concept of circadian rhythm, misalignment, and its connection to metabolic syndrome. When the circadian rhythm becomes desynchronized, it is akin to an orchestra playing out of tune. So I delivered this talk at the 33rd Annual Convention of Diabetes Philippines, which took place at the EDSA Shangri-La Hotel on November 11, 2016. Okay, in this presentation, I'm going to undertake the following topics. First, an introduction, a brief history of circadian time, often referred to as the rhythm of life. And then I'll go to the most challenging part of the presentation, the mammalian core clock gene and how this system works. I will try my best to simplify it based on my understanding. The next is a circadian clock in health, harmonization of internal clock, and how do circadian affects the body function and health, and how this internal clock synchronizes with the external environment. Then what happens when this is misaligned? What are the causes and consequences? And finally, go to my summary and conclusion. So let's start with the basic things here, and that's the Earth rotation. As the Earth orbits around the Sun, the Earth pirouettes on its own axis, so that part of the Earth surface is alternately exposed to sunlight and darkness. For nearly 4 billion years, life on Earth, outside of the North and South Poles and deep oceans, life on Earth has evolved under a consistent pattern of alternating bright days and dark nights. As a result, most organisms of our planet develop an internal timekeeping system, the so-called built-in internal body clock, to synchronize with the outside light-dark cycle, which allows the organism to assign function based on the time of the day. Therefore, there is always best time of the day physiologically to do our activities such as exercise, study or work, eat, sleep, or even copulation. Now, and this cycle or oscillation is called the circadian rhythm. The word circadian came from Latin circa, meaning around, and dies, meaning day. So when you combine the two, it means approximately 24 hours. Today, the term is used to signify biological events that predictably reoccur once every 24 hours. The graph here shows an example of two important hormones, melatonin and cortisol, that oscillates during different times of the day. Melatonin level is highest at night, which is considered as the sleep hormone, and the reverse is true with cortisol, which is highest level in the morning. In short, circadian rhythm is important to an organism. Why? Because number one, it allows the organism to anticipate rather than to react with the daily changes in the external environment, and to synchronize their behavior and physiologic processes. These oscillations is also help an organism predict environmental changes in order to optimize energy utilization, reproduction, and survival. The Center for this circadian biology is a relatively new field of science known as chronobiology. The word derived from Greek. Chrono means related with time, biology means science of life, and logos means study. It is therefore the study of science of life in relation to time. Chronobiology studies includes but are not limited to chronophysiology, chronopathology, and chronopharmacology. While chronophysiology study of time on physiologic process, while chronopathology deals with the subject of disrupted timing in vital biological processes, and chronopharmacology deals with the subject deals with the effect of drug therapy, which is very important and clinically relevant because some enzymes in the body undergo significant diurnal pattern, like for example hepatic HMG-CoA reductase, very important in the synthesis of cholesterol, is active at night, so that it's recommended the best time to give statin is at night. Now, the first hint that daily rhythm are internally driven in the cell was from the experiment done by Jean-Jacques Demarian in 1729, a French geophysicist, astronomer, and a chronobiologist rolled into one. He noted that 24 hours pattern in the movement of the leaves of the plant Mimosa pudica, lowering of their leaves at night and raising it back in the morning. This oscillation pattern continued even when the plants were kept in constant darkness. Thus, this was the first experiment to attempt to distinguish an endogenous clock that may or may not be influenced by external environment. So the internal clock does not only exist in plant but also animals. When you place a mouse on a running wheel and record their activity profile, that is then captured in this octogram, looking on the graph, the top bar represent LD or light dark cycle, white bar represent 12 hours of light, and black bar represent 12 hours of dark cycle. The gray bar represent when the animal begins their activity. As you can see, mice are nocturnal. They are very active in the morning and they rest at night. When they are exposed to light dark cycle, animal predictably begins their activity approximately the same time for a week. However, when kept on a constant darkness for another week, the animal continues to have rhythmicity. However, the rhythmicity is out of phase. The animal beginning to run earlier each day, succeeding days. As you can see, the gray bar is shifted to the left. So that these two experiments tell us, number one, that each individual cells had its own self-sustained clocks, and number two, these internal clocks are synchronized by an external time cues, or the light-dark cycle. Even without these external time cues, cells continue to oscillate so that rhythmicity persists. However, rhythmicity becomes out of phase and no longer aligned with the light-dark cycle. So from Jean-Jacques Demarian experiment in 1729, we go fast forward in the timeline. And in 1994, it was this guy, Professor Joseph Takahashi, a Japanese-American neurobiologist and geneticist, professor at University of Texas Southwestern Medical Center, discovered the first mammalian clock gene. The mammalian circadian clock gene consists of a series of a complex interlocking transcription, translation, feedback loops. You can see the complexities of this slide and it will take us probably forever to completely understand all of this. So the audience is advised, if interested, to read some excellent review articles, including the articles cited below. The clock gene is not far from the complicated Tokyo subway map. What makes it even worse is all the streets names are foreign sounding. This is also true with the terminology with the mammalian clock genes. But I need to put up few slides to discuss the clock genes, so we will appreciate it more or better, especially when I start linking it to metabolism. For simplicity, just remember clock genes are genes of twos. There are two transcription factors, namely clock and BMAL-1. Long name of clock and BMAL is there, but that's not the most important. There are also two transcribed proteins, namely PER and CRY. PER refer to period, CRY refer to cryptochrome. And lastly, there are two regulatory loops, which are the core loop and the auxiliary or supplemental loop. Okay, so with this, it's quite easy to remember the human clock genes. Now, let's go to a more challenging one and understand how this system works. But before everything else, let me orient you with these cartoons. Inside the circle is the nucleus. Any activity inside the circle is activity occurring in the nucleus. And outside of the circle is the cytoplasm. Now, transcription factor clock and BMAL, that's one, heterodimers, and then complex bind to a promoter gene, also called EBOX, of period and cytochrome gene, better known as PER and CRY genes. This leads to transcription of transcription of messenger RNA, and this messenger RNA then exported to the cytoplasm from the nucleus for PER and CRY proteins translation. Once the level of PER-CRY becomes adequate, it then undergoes post-translational modification by phosphorylation. This leads to this time heterodimerization of PER and CRY, and these complexes are then transported back to the nucleus. The PER-CRY complex in the nucleus bind to the clock BMAL, thus one, then inhibiting PER-CRY transcription. As the PER-CRY level decline because of degradation, the inhibition is removed or lifted, and the cycle starts all over again. So, in summary, the binding of clock BMAL transcription factor complex to the EBOX and transcription and translation of PER and CRY comprises the positive component of the loop, while the PER and CRY complex is the negative component of the loop. That is why this is called transcriptional translational feedback loop. This entire feedback loop takes approximately 24 hours. So if you look at the level of period or PER protein, it oscillates. It goes up and down over a period of 24 hours. It is in tune with the light-dark cycle, lowest at the midnight and rise up in the morning and reach its peak at midday and then fall back down in 24-hour basis. Another regulatory loop is induced by clock BMAL, thus one heterodimers, activating transcription of two orphan nuclear receptor. Clock BMAL, thus one, activates transcription of both REV-ERB and ROR, or retinoic acid receptor related orphan receptor alpha, and these two transcribed proteins that binds with the ROR alpha and REV-ERB alpha and regulate BMAL thus one activity. REV-ERB repress, whereas ROR activates BMAL thus one transcription. So in a big picture, looking at both regulatory loops, that is in the upper portion in green, and the accessory loops, the lower portion in red, and in the middle is the transcription factor gene. The transcription factor complex activate transcription of per and cry proteins, and the per cry complex translocate back to the nucleus and inhibit clock BMAL activity and inhibition of translation of protein. When the level of these two proteins drops, the translated protein per and cry drops because of degradation. The inhibition is then removed or lifted and the cycle starts all over again. Normally, the core loop is very efficient. However, sometimes when it malfunctions, that leads to abnormal activity and level of BMAL becomes abnormal. Then the accessory loop takes over and keep the system checked. But how? Abnormal level of BMAL, activates two orphan receptors, and that is in red, and depending of the level of BMAL does one, when it's high, Reb alpha repress, and when it's low, ROR activates the clock BMAL 1 transcription factor. Through this discovery of mammalian clock genes, scientists found out that virtually all human cells have their own self-sustained internal clock. And experts, by the use of DNA arrays, believe that about 10 to 30% of genes in the body were under clock control. You can see clock are everywhere. It is found in brain, lungs, liver, skin, fibroblasts, etc. Now, talking about skin fibroblasts as a proof of concept that virtually all individual cells of human have their own self-sustained internal clock, human skin fibroblasts taken out from the body and kept in culture display robust oscillation, as seen here with individual cell oscillation analysis. However, over time, individual cell becomes desynchronized and have slightly different periods. So, because of this synchrony, this internal clock have to be finely tuned by the circadian system from the outside world or the environment to maintain a healthy status. It is likened to a musical orchestra. When playing a classical tune, all instrument has to be harmonized to give you a perfect and harmonious sound. The question is, how are these different internal clocks in the body kept in perfect harmony? It turned out that there is a master clock. The discovery of clock genes enabled the identification of brain that contains the molecular machinery necessary for generation of circadian rhythm. It is situated in the suprachiasmatic nuclei, a tiny region of the brain in the hypothalamus, that just above the optic nerve. This suprachiasmatic nuclei gets its cues from the light that hit the photoreceptor and the retina. And light is the most important Zeitgeber. In German word literally means time giver. Then sent this information on the other areas of the brain like the pituitary and pineal gland and other parts of the hypothalamus. Hormones such as melatonin and cortisol are released into the bloodstream to control physiologic and behavioral responses such as body temperature, blood pressure, among many others. Environmental cues such as eating, activity, and body temperature can also reset the clock. These are the list of neurohormones and neuropeptides that serves as connection between the central and peripheral clock. These are the various Zeitgeber that can modulate this transcriptional pathway and reset the clock. And light is the most important size givers. Okay, just to emphasize the different environmental cues such as light, food, and temperature can reset the body clock through multiple pathways. These cues synchronize with the peripheral tissue like the liver, skeletal muscles, adipose tissues, and pancreas for glucose metabolism and other metabolic processes. There are three basic components of the system. The environmental cues, in which light is the most important. The center is the central pacemaker, present it to the brain's suprachiasmatic nuclei, which receive the input and send it out to the peripheral clock to regulate the timing of various processes such as behavior, sleep-wake cycle, and metabolism such as feeding, fasting, and body temperature rhythm, and etc. A perfect harmonization or synchronization of circadian rhythm is important to keep us healthy. As mentioned before, just like a conductor for an orchestra who harmonizes sound coming from all the individual instruments. However, when disruption of circadian clock occurs, then the orchestra is out of tune, leading to disharmonization of sound coming from the different instruments. So that the health implication of disrupted circadian rhythm. Number one, for many thousands of years, the sun was our only source of light, and human behavior followed a natural day-night cycle. This environment began to change approximately 150 years ago with the invention of incandescent lighting by Thomas Edison. While his invention undoubtedly solved many of our problems, however, he made people less reliant on the sun's natural rays, so that light becomes available at all hours of the night, and thus resulted in the disrupted circadian clock rhythm. Moreover, modern technology is now keeping us up later into the night and cutting our precious sleep. As a result, many of us are sleep deprived, and sleep deprivation is associated with a gamut of disorder affecting our cognitive function, alertness, metabolism, musculoskeletal system, cardiovascular, and even our immune system may be affected. In 2003, U.S. National Sleep Foundation survey showed that around 30% of adults report sleeping less than 6 hours per night as opposed to 9 hours a century ago, and this extended time of being awake may be testing the body's limit to maintain metabolic and hormonal equilibrium. Circadian disruption may arise from genetic, gene mutations, environmental, such as shift work factors, and contributes to the development of behavioral and cardiometabolic disorder and even cancer. For this presentation, I will concentrate on the cardiometabolic disorder. Now, there are three lines of evidence linking the circadian desynchronization to metabolic syndrome. Number one, evidence from the experimental animal model with the use of mutant clock genes. Number two, epidemiological studies, which is an observational studies. Usual subject use are the shift workers or night shift versus day shift workers. They were observed for months to years and evaluate for any ill effect of disrupted circadian rhythm. And number three, an experimental human studies. These are usually normal healthy individuals. These studies creates an acute abnormal condition by reducing hours of sleep for few days and individual allowed to recover, what is called sleep deprivation or death and sleep recovered. Now let's go now first with the clock mutant mice studies. These are animal studies. So homogeneous clock mutant mice have a greatly attenuated diurnal feeding rhythm. They are hyperphagic and obese and develop a metabolic syndrome of hyperleptinemia, hyperlipidemia, hepatic steatosis, and hyperglycemia with insufficient compensatory insulin production. The level of hypothalamic peptide expression associated with energy balance were greatly attenuated in clock mutant animal. Now, taken together, disruption of this circadian network can lead to obesity as well as metabolic syndrome in mice. Now, looking at the data, clock mutant animal fed with either a regular or high-fat diet showed a significant increase in energy intake and body weight compared to wild-type control. Shows the time course for the weight gain in clock mutant and wild-type adult animal fed either a controlled or a high-fat diet for a period of 10 weeks beginning at 6 weeks of age. You see a significant weight increase. First 8 weeks of life, meaning newborn to 8 weeks, the significant weight increase started to be obvious at 4 weeks, suggesting the mutation did not affect fetal growth or nutrition. And when they look at time course, the obvious changes between wild type and mutant mice in diurnal rhythm and the PER2 messenger RNA and selected hypothalamic peptides involved in energy balance. The red is the wild type and the black curve is the clock mutant. When they look and measured metabolic parameters, which show significant increase in triglycerides and leptin, but no significant difference in the level of insulin. Now, let's go now to the second group of studies, the evidence from epidemiological studies. Shift workers. The following epidemiologic studies tried to evaluate or establish the association between shift workers with disrupted circadian rhythm and metabolic disorders. Let's try to define what is a shift workers. Okay, shift works has become more prevalent during the past decades and show clear negative effect on sleeping, timing, length, and quality. Shift workers are active during the night and sleep and eat at abnormal hours. They fall asleep in the morning but are awakened ahead of time owing to their circadian rhythm. These epidemiologic studies try to establish the association between shift workers with disrupted circadian rhythm and metabolic disorders. The study by Petrosti, an Italian study looking at the incidence of metabolic syndrome among night shift healthcare workers in Vergala University from Institute of Occupational Medicine. They are male and female nurses performing night shifts free from any component of metabolic syndrome at baseline were evaluated annually for the development of disorder during a 4-year follow-up. Male and female nurses performing daytime work only visited during the same time period represent the control group. You can see very early separation of the curve, less than 1 year, okay, between daytime worker and night shift worker. Another study, this time a meta-analysis done by Wag et al., they took 13 eligible articles, all observational epidemiological studies about night shift workers and metabolic syndrome from the database, including 3 cohort studies, 1 nested case studies, and several cross-sectional studies. The pooled cohort and nested case control and overall increases in the relative risk of metabolic syndrome in shift workers at about 43%. Same is true on the cross-sectional studies, a 36% increase in the metabolic syndrome. Furthermore, shift workers has also been associated with increase in vascular event as seen in these observational studies published in the British Medical Journal. Now, another epidemiological studies on sleep duration and metabolic disorder. Insufficient sleep are associated with overweight and obesity. Another study looking at sleep duration as a risk for development of type 2 DM, a significant U-shaped relationship between self-reported sleep duration and the incidence of type 2 DM. Okay. 7 hours, the age adjusted, it's just 1. If you sleep less than 7 hours, the risk increases. However, if you sleep more than 8 hours, there's a U-shaped curve leading to increase the incidence of diabetes. Now, we go to the last group of evidence. This is from human experimental studies. The study looking at sleep curtailment and hormones that affect the appetite, which is leptin and ghrelin. Normal, healthy individuals cut their hours of sleep for a few days and measured their leptin and ghrelin and the rate their hunger and appetite. The data shows in 12 healthy men restricting their sleep for 2 days and extending the sleep for 2 days measured leptin and ghrelin level. The leptin and ghrelin is believed to represent the yin-yang of energy balance. Leptin considered as an anorexigenic effect, and conversely, ghrelin stimulate appetite. The mean leptin level were 18% lower when sleep was restricted, and the mean ghrelin level were 28% higher compared to sleep recovery condition. Rating of hunger and overall appetite were also increased in sleep depth individual. Sleep depth yellow and sleep recovery is blue. Another human experimental study by the same author, a much earlier study, evaluating the impact of sleep depth not only on metabolic but as well as endocrine function. The rate of glucose clearance were nearly 40% slower in a sleep depth condition than in the sleep recovery condition. The acute insulin response to glucose was 30% lower in the sleep depth condition than in the sleep recovery condition. A decrease of acute insulin response to glucose is hypothesized as an early marker of diabetes. Sleep-deprived individual does not only affect glucose metabolism but also other hormones such as cortisol, growth hormone, leptin, as well as autonomic nervous system activity. So clearly, sleep depth is associated with metabolic and endocrine alterations, and that may have pathophysiological consequences in the long term. Last study is derived from the Wisconsin Sleep Cohort Study, a large longitudinal population-based study on sleep disorder. The study revealed that chronic sleep deprivation, sleep less than 8 hours, was associated with increase in BMI, a U-shaped curvilinear association between sleep duration and BMI was also observed. So while the association between circadian disruption and metabolism is strong, the underlying mechanism that leads to the development of metabolic disease, obesity, diabetes, and cardiometabolic disorder remain poorly understood. As discussed, disruption of circadian rhythm is strongly associated with metabolic syndrome. Metabolic syndrome is composed of several metabolic abnormalities, including central or intra-abdominal obesity, dyslipidemia, hyperglycemia, and hypertension. This syndrome has become a major public health challenge worldwide. The main driving force for the worldwide epidemic of obesity and metabolic syndromes can be traced with the excess food intake and physical inactivity, more in industrial nations than in developing countries. In addition, mounting and emerging evidence from the clinical epidemiological studies that I have already mentioned has led to the hypothesis that one of the major changes in the industrialized world that contributes to the pathogenesis of metabolic syndrome involved introduction of artificial light and work into the night time. In addition to the pervasive rise in voluntary sleep curtailment, indeed, this common disorder of circadian behavior and sleep and associated with increased hunger, decreased glucose and lipid metabolism that eventually leads to obesity, metabolic syndrome, and cardiovascular disease. So, living organisms on Earth are under the steady influence of daily and seasonal changes resulting from a planet rotations and orbit around the Sun. This periodic pattern, most prominently manifested by light-dark cycle, has led to the establishment of endogenous circadian timing that synchronize biological function. The mammalian circadian system is composed of many individual tissue-specific clocks and orchestrated by the master circadian pacemaker residing in a suprachiasmatic nuclei of the brain. There is growing evidence to suggest that desynchronized circadian rhythm is a risk factor for the development of metabolic syndrome. In our modern society, individuals are voluntarily decreasing sleep duration for various reasons without knowing its future impact on their health. Better knowledge in chronobiology may ultimately lead to improved therapeutics for metabolic syndrome as well as cardiovascular disease. So, my final slide to keep up with the title of my presentation. While circadian rhythm that is synchronized with external environment leads to a normal metabolic function, circadian disruption, on the other hand, impair metabolic function and leads to metabolic syndrome. Therefore, a desynchronized circadian rhythm is similar to a musical conductor that miscommunicates with their musician either by a wrong stroke of button and other gesture that leads to this harmonious musical sound. And for that, I'll bring my presentation to a close, and thank you for listening.